So that's all the Korean I can offer. The rest will be in English. Before I start, let me first thank you for the invitation and to the opportunity to give a talk here. Before I talk, I would like to know where you are and who you are. So who is from KAIST? Just raise your hand. Who is from KAIST? So a few. Who is from other Korean universities? Okay, who is not from Korea? You are and you are. Okay, where are you from?
There's one more from Japan.
From Japan? France? Okay, and Japan as well. Yeah, wonderful. Now, who is a graduate student? Who is a graduate student? Raise your hands. So the clear majority. Okay, so the level of my talks will be clearly directed to graduate students. And I invite you to ask questions whenever you want. So whatever seems kind of strange, fire away a question. You can also ask in Korean if this is easier for you, and then you will translate for me. So here on the homepage of the course of the workshop, I have the lecture notes here as number 1 and number 2. And the first maybe about 6 lectures or so will be based on the first manuscript. Diffusion limit of transport equations in biology with Amanda Swan. And then I'd like to point out this little citation here. A smart model is a good model. Of course, I relate this to mathematical models, right? A smart model is a good model, or a good model is a smart model. But this was actually said by Tyra Banks, who is a fashion model. So she was she was making a very different statement, and I took it out of the context. So how do I get back? How do I get back?
Just click there.
Click where?
Escape.
Escape? No. Yeah, that's good. Yeah.
Or just escape. Or chapter 11.
Okay, so now I'm good. Yeah. Yeah, so Yeonjong said I give 8 lectures, and of course there's lots of material, so here's a little outline of what I'm going to do. So today I talk about, I introduce what I like to talk about. We'll talk about the random walks, very simple random walks, and introduce chemotaxis. Now, who of you have heard about chemotaxis before? So who have heard about chemotaxis? So if not everybody raises a hand, then you have not done a good job. So I will also today introduce chemotaxis, not do any theory, just about the modeling, what it means for the biology. And then tomorrow we really start with the meat of things. I start in 1 dimension with a correlated random walk in 1 dimension, because in this case we can do many things explicitly. We can write down the equations, we can write down the transformations I do, we can write down the scalings I'm looking at, etc. And all this can be understood more or less completely. But then I would like to lift it in number 3 for transport equations in higher dimensions. And then we really need some advanced mathematical theory, like Krasnodębski's theorem and Krein-Rootman theorems and stuff like this. Talk about the turning operator, the Pearson walk, movement and fiber network. And then basically the highlight of all these lectures is the diffusion limit. So I would like to formally derive the diffusion limit, but then also mathematically find some properties. For example, the structure of the diffusion tensor, which can be anisotropic or isotropic. We can have persistence or no persistence. So I will introduce all these terms. And then 7 and 8, it's basically towards applications. So in particular in 8, I will talk about applications to brain tumors, to wolf movement, and then the sea turtle navigation I'm taking probably out of this lecture, because I'm going to talk about next week at NIMS about the sea turtle problem. I know several of you will not be there. Let's see how the time goes. Maybe I can sneak in some of the sea turtles as well. But I think the most interesting example is actually the brain tumor growth. And so everything we do now, we work towards the model for the brain tumors and see how mathematics can help us to maybe understand and help treatment of these tumors. Okay. So, but before I really start, so I'm interested in mathematical biology, so whatever PDEs I look at, they should have something to do with reality and something to do with biology. So I like to start to actually talk about measurements, to start on the biological side and see what kind of data do we get from biologists. So I'm not doing experiments myself, but I certainly can read papers where experiments are done. And so it's interesting what kind of data do we have, and then start from there to talk about mathematical modeling. So if you hate statistics, then don't worry. I will not talk about inference and all that kind of stuff. But still, I would like to set the background. So, introduction. One measurements. So talking about kinetic equations, transport equations, reaction diffusion equations, I'm interested in cell movement or species movement. It could be a wolf, it could be cells, it could be something. So I'm interested in some movement properties of species. And a problem which we have to address is we have individuals which move, individual movement. But we would like to say something about a population movement. Okay, so for example, a tumor, if you have a tumor and the tumor is spreading and metastasizing, reaching other parts of the body, individual cells move, but what you don't see the individual cells moving, you just see a tumor growing. So there's a connection between individual movement and population movement, which I like to make. Okay. Now measurements, often measurements are done on the population level. And so then let x of t denote a random variable for the location of an individual. Yeah, individual at time t. But rather than being able to follow each cell individually, we might only see the population. So what we see would be A1 like the expected location or the mean location. So the expectation of x of t, and in some cases we can also get the variance of it. So basically the uncertainty of the location. So A2 would be the variance of the location, Vx of t. Okay, so these would be measurements on the population level. You have many, many individuals, and you just see what they do as a population, as a whole. However, in modern biology, we are more and more able to measure individually as well. So individual measurements. So for example, cells, there's now something like confocal microscopy where you can indeed follow individual metastatic cancer cells and see how they move through tissue, what kind of tracks they make. If you look at wolf, I mentioned wolf over there, you can have GPS collars on the wolf, and then you can measure where the wolf is every 5 minutes, you get a signal. You can do this also for dolphins, and you can do this for many, many species. So you follow maybe a track, which can maybe look like this, where an individual moves and changes direction, moves around. Maybe you do not get a smooth curve, you might only get selected data points in certain time distances. But still, so what you can measure is a mean speed of the species. So I call this gamma. B2 is a mean turning rate. Which I call mu. And then B3, so the turning rate tells us how often do they change direction. And then the final one is which directions do they choose. Right? So it could be that they always choose going to the right or something. So then this is a turning distribution. So distribution of new chosen directions. So T of V prime where v and v prime are velocities of the individuals. So we have two sets of measurements, and they are conceptually very different. One is on the population level and one is on the individual level. And then the natural question is, of course, how are they related, right? So how does one lead to the other, or one to the one? So the big question is, what does one have to do with the other? And the answer is basically number 5, or lecture number 5, when we do the diffusion limit. Because so we get there, but it takes a little, it takes some work to get there. So today, Monday, I will talk about part A, and then Tuesday, tomorrow, I will talk about B, and Wednesday I talk about B, and Thursday I talk about B, and Friday I talk about B. So it's not evenly distributed, as you see. Okay. So let's start with individual measurements, 1.2, and let's look at a simple random walk. Random walk on a grid. Here is x, and here is time. Now I really start very, very simple, so I'm sure many of you are quite familiar with these kind of random walks. I think still it makes sense to start on the easy level and then progressively get more complicated. On the other hand, it might be new to some of you, so that is fine as well. Okay, so I use a grid here on x, which has a size delta, and a time step which I call tau. And I'm looking at a random walker which can jump left or right. So let's maybe I start here, and then I jump to the right with a probability q, and I jump to the left with a probability 1 minus q, meaning that there's no probability of staying where you are, so you either jump right or you jump left. That's it. So we have an individual that starts at 0. So then I better call this 0 here. And then q between 0 and 1 is a probability to jump to the right. And then, of course, 1 minus q is the opposite probability to jump to the left. And as I say, we do not allow it to pause or to stop. We can include a probability of resting, and the same thing works fine as well. But this is just a little bit simpler. Before I go on with this, I give you 2 references. One is the book by Sauderer, PDEs. It is a classic textbook on PDEs in North America. I don't know if it's so well known here in Korea. But there, this kind of random walk ideas are laid out very nicely. And the other one is a book by Okubo and Levin, 2004. It's on diffusion and ecological problems, and I can highly recommend this book. So many of the aspects we are discussing here I also discussed in Okubo and Levin's book. Okay. So is there a second blackboard behind it or not? No. That's it.
There's a whiteboard.
Oh, I use the whiteboard, sure. Let's see if I have enough pens. Can you still see it from France? Very good. All right. So what I called xt was the time random walk for our random walker. Now I have a discrete time step n, so I increment by always by 1. So xn now is the location of the random walker at iteration number n. And so now I'm interested in the expectation. And so the expectation after one jump. Where do I find my particle after one jump? Okay, and so then I just use the definition of an expectation. So the sum over all y probability x is equal to y. And I can only jump right or left. So then I can figure this out. So this is delta times the probability q plus minus delta times the probability 1 minus q and then I can simplify this together and I get an interference here. Where is this? Okay. So maybe I'm just on talk when I stand on this location, then I can have no interference. So this is then delta 2q minus 1.
The q is uniform in every position?
Yeah, very good question. So q is the same in every position. Yeah, so very simple. And then after n steps, we get basically this expected location, 2q minus 1, plus the expectation of n minus 1 jumps, and so on. So we get a recursive relation for this expectation. And in the end, it's just n times what we had before, 2q minus 1. So what's happening here already is part of the answer for my measurements A1. So A1 was I can measure the mean location of my particle, and here is the mean location of my particle, and I get something which involves delta and q. So I have already one information for one of these parameters here. Okay. So let's look at this just quickly. So if q is 1/2, which means we have equal probability to jump right or left, then the expected location is 0, right? So this term is 0. So then E xn is 0. If q is bigger than 1/2, so then we have the expected location is, q bigger than 1/2, so this term is positive. So the expected location is positive, so we are jumping to the right. We are jumping more to the right than to the left. So the expected location is to the right. And if q is less than 1/2, then of course the opposite. Xn is less than 1/2, less than 0. Okay. Similar for the variance, maybe you can do that here too. Variance of xn is expectation of, let's do x1 first. Expectation of x1 squared minus the expectation of x1 all squared. And then you can compute this out to be 4 delta square q, 1 minus q. Again, right, you can, for this expectation of xi square, you can use this formula with y square and then sub everything in and compute it out. And then the variance for n independent jumps is just n times the variance of x1, which is 4n delta square q, 1 minus q. And then we use the second information. So if we have information about the variance of our random walker, then we can use it here. And then indeed we can estimate maybe 2 parameters q and delta from here and here. Okay, so then we can describe the model or can relate the model to the data. Let me write this down just quickly.
Do you want to use the whiteboard also? There is another one behind the...
No, that's fine. Yeah. Right. But now I cannot use this pen, right? This will go wrong at some point, I'm sure. Okay, so I have these guys. Yeah, so now I would like to write this in continuous time. So t is n times tau, so I just have time step tau and I use it n times. And this gives me my t because I would like to replace the n which I have here and here. I replace it by t. So then n is t divided by tau, and then we get, what do we get? Yeah, so then I can define a mean velocity, which I call c, and so this is then the expectation divided by t. Okay, so I get take the formula for the expectation from here. I replace n as t over tau, and then I divide by t. So the t disappears. And I get delta divided by tau 2q minus 1. And this is an expression I'm going to use later. So that's why I introduce it here. And this has a meaning of a mean velocity. And I introduce something like a diffusion coefficient. Which is d 1/2 of the variance, xt divided by t. And then if I sub this in, I get 2 delta square divided by t, by tau, q times 1 minus q. Okay. As I say, these are quantities which show up later, so it makes sense to introduce them already here, right now. Good. So now I talked about one random walker on a grid. Now I would like to raise the level and talk about a population of random walkers and look at the distribution of random walkers in the same situation. So we use a population model. And so I introduce P of xt to be the probability density of a population of independent random walkers. We'll just see the time. Okay, that's good. And so here we use the master equation. So that's a very common technique for stochastic processes. Master equation. So the probability to find an individual at x and time t plus tau is given by q times the probability that it was at x minus delta a time step earlier, and then jump to the right, and jumping to the right is q. Okay, and then plus 1 minus q probability of being at x plus delta at time t, and then jumping to the left, and the jumping to the left probability is 1 minus q. So this is called a master equation. And a very common method is to use a Taylor expansion of this master equation for the case when tau is small and when delta is small, so then we can throw away higher order terms. And it is very good if everybody in the world has seen this once. So I show this to you once now, and then you have seen it. And many of you have seen it anyways, I'm sure. So just quickly, Taylor expansion. So P of t, what do we have? P of x and t plus tau is then P of xt plus tau times the time derivative pt of xt plus tau square over 2 ptt of xt, and so on. And I use the index notation for the partial derivatives. I think most of you are familiar with that. And then this equals now the right hand side. So we have q times p at xt, and then this is minus delta, I guess, minus delta px plus delta square over 2 pxx, and so on, plus 1 minus q p plus delta px plus delta square over 2 pxx, and so on. And I left away the arguments x and t, but they're all evaluated now at the point xt. Okay. So let's see what the right hand side gives. So the p term, we have qp and 1 minus qp. So together this is just p. And then for the delta term, we have a minus q delta. And here we have a minus q delta, and we have a delta. So then this is delta times 1 minus 2q times px. And then the delta square term, it has the same factor in front, so then this is delta square over 2 pxx plus higher order terms. Okay. Yeah. And now the P and the P cancels, this guy and this guy. And so then the leading order term is a term which has a factor of tau and a factor of delta. And everything else is higher order. Well, almost. So let's see. So I keep the following terms. Leading order is then PT, and I divide by tau. I divide by this factor tau, so then I get delta over tau 1 minus 2q px plus delta square over 2 tau pxx plus higher order terms. Oh, no, I said leading order, so then throw this away. Okay. So now, somebody might notice something compared to what we did just before in section 1.2, say. Yeah, tell me, what do you notice?
There's a hint at the left side.
The hint at the left, oh, it's right here, yeah. Right, so this term shows up exactly, right? So this term is exactly the term in front of px. Well, with a minus sign. So this is minus c. Now with the diffusion term, it is not exactly this d here, so we have to divide by this q1 minus q. But that's fine, so then this here is d divided by q1 minus q. So what we get from this Taylor expansion is an advection diffusion equation, right? We have a diffusion term here, and we have an advection term here. If we put it on the other side, we get plus c, so it has the right sign. It's all nice. But still, we have all these deltas and taus and q's in there. So what we can do now is look at different scaling limits, and different scaling limits give us different results. And that's important. So, different scaling limits. And I'm going to show you, well, 2.5 or maybe 3 scaling limits. We can do an infinite number of scaling limits, and they're all useless, except those 3 which I'm going to show you. Okay, so what do I mean? I'm interested in the limit when delta goes to 0, when tau goes to 0, and then also when q goes to 1/2. And now I have 3 variables which I send to 0, and I can do it in very different ways. Right? This is basically a limit going to 0 in R3, so you can approach 0 from any whichever way you want. So let's start here. So let's say the ratio delta over tau converges to an alpha, which is constant. Okay. So then here I have delta over tau. This goes to a constant. Here I have delta square over 2 tau. So let's look at this term. So then delta square over 2 tau is delta times delta over 2 tau, which goes to 0. So this term goes to 0, which meaning the diffusion term disappears and the drift term is maintained. So then we have pure transport equation so pt plus cpx is equal to 0, and that's it. Okay, and so that's a very simple equation. It means drift dominated, so the particles are just ballistic. They just go in their speed, with their speed in their direction. They don't change, they do anything, they just go. So that is limit A. B would be, so what about the other case? If you use this term delta square over tau and send this to become a constant in the limit. So assume this goes to what? Constant for delta to 0 and tau to 0. Yeah, if I do this, then let's look at the first term here. But now the first term involves a q, And so now I have to say if Q is 1/2 or Q is not 1/2. And so these are two subcases. So B dot 1 would be Q is not 1/2, where then the term delta over tau is the same as delta square over tau times 1 over delta, which goes to infinity. Okay, so delta goes to 0. Delta square over tau is constant, so this together goes to infinity. So this term would be unbounded, and we get no useful result. So this doesn't work. And now the last case, maybe I put it in here. So then B2. So what if Q goes to 1/2 at the same time? So if Q goes to 1/2, then this term goes to 0 and can kind of compensate from the bigness of everything else. So it has to be done in the right way, so such that delta over tau 1 minus 2Q converges to minus C. So in this limit now everything goes to 0. Delta goes to 0, tau goes to 0, Q goes to 1/2. Everything fine-tuned exactly so that I get minus C out of this. So maybe I write this down here. Tau to 0, Q to 1/2. So it looks very artificial. If you see this the first time, you think this never is going to happen. This is very artificial. But this is in fact the basis of most advection diffusion equations. So that the advection and diffusion scale in exactly the right way. So what do we get in this limit? Oh yeah. So this term goes to minus C, and then we have still this term D divided by Q, 1 minus Q, and a 4. Why the 4? I forgot the 4. So here must be a 4. I forgot this term. Because we divide by 2 tau, and we have another factor 2 from here. So then we have a 2 together, a factor 4. That's important now, because if Q goes to 1/2, then this is 1/2 and this is 1/2, so it's a quarter, and then the 4 cancels. And so this in this limit goes to D. So tau to 0, delta to 0, Q to 1/2. So the first term goes to minus C, the second term goes to D, and now I can piece it together, and we get an advection diffusion limit. So together we find PT plus CPX is D PXX. So advection diffusion equation. This is an advection diffusion equation you see a lot in your PDE studies. But not only did we derive this now, we also relate C and D to the measurements, right? So C is still given by this, and D is still given by this. And these were the measurements of our population which we had. Okay. Yeah. All right. Okay, so questions about this, as far as I got. Yeah.
One question?
Yes.
I understand that we mathematicians love to take limits.
Yes. Mathematicians love to take limits. Yeah, exactly. Yeah.
How if you have that equation, then?
So this one, yeah.
Yeah. Instead of taking limit, just stop there. How about not to take the limit?
Yeah, but then you have.
If whatever the delta and tau is given,
Yes.
then the coefficient exists something like that.
Yeah, but then you have the higher order terms, right? Then you would not like to keep. So then you what do you get? You get third order derivatives, fourth order derivatives, and so on. Yeah.
The coefficient is smaller.
And the coefficients are smaller, right? Yeah. Yeah.
And I also So in that way, the last case you said that taking that limit looks artificial. That's why you just said, but that's the way it always works.
Yes.
Right. So what I feel is well, actually that one is to keep this coefficient.
Yes. Right.
Okay.
yeah, that's exactly right. Other questions about this bit? Yeah.
Why can we ignore the second derivative of p with respect to time, ppt?
A very good question, yeah. And so we cannot, or we can. So it's about this term. So in these limits which I considered there, you will see that the tau square term goes to 0 very quickly. So that is a higher order term. But it does make sense, so I have to contradict what I said earlier. It does make sense to consider limits where the PTT term also survives. And then you get telegraph equations, and they can also be studied. Yeah. So that was a very good question. But I'm not going to do this now. I might do this tomorrow, actually. Okay, so now I would like to use this for chemotaxis. So now let's come to chemotaxis and talk about that. So 1,4 chemotaxis. Yeah, what is chemotaxis? So chemotaxis has to do with cell movement, and then cells secrete a chemical signal, and other cells can smell the signal and move up gradients. And then this leads to aggregation, so that cells come together because they release kind of an attractive signal which invites other cells to come in. And I would like to model this on the diffusion kind of level. So I would like to have a model right on this level here as an advection diffusion model. So let's look at the diffusion equation first. And we have seen this in Dr. Matano's talk as well. So ut is duxx, just in one dimension. Yeah, now I'm switching dimensions, so then I do it in higher dimensions, d delta u. And then I would like to look at the flux. So the flux is, in this case, it is just minus d gradient u. So the assumption is the flux is proportional to the gradient of the population, but with a minus sign. So wherever the population is big, the flux is in opposite direction and tries to equilibrate everything. And this is also known as Fick's law. Fick's first law. Okay. Now, I told some of you that I'm from Germany, and the word Fick has a very specific meaning in German. It means having sex. So German students never have a problem to remember this law, just because of the name. So anyway, so this is Fick's law. Well, now let's extend it to include chemotaxis. And so this was done by Keller and Siegel. Keller-Siegel model 1970. And we have a chemical signal. Triggers a chemotactic flux. Which is proportional to the population. So then we have J chemical is just beta times U. So meaning the more cells you have following the chemical, then the bigger the flux is. But then we need something for beta. So then the next assumption is so the rate beta depends on the signal. On the signal S, let's call it S of x and t, and its gradient so gradient S of x and t. So if particles go to high concentrations of S, meaning they follow the gradient of S. So this beta actually is a vector, right? So J is a vector, beta is a vector, U is a scalar. So this beta is in direction of gradient of S. So we write this as beta is he of S gradient S, and then he is called chemotactic sensitivity. All right. So then let's put the fluxes together. So let's assume we have a diffusive flux and a chemical flux given with this beta term. So then the total flux is J is minus D gradient U plus he of S U times gradient S. And now if we go back to the diffusion equation, so then we plug in the flux into the diffusion equation. So we have UT is minus gradient of JU, which then is delta D delta U minus he of S U delta S. Okay, and so this is the first part of the classical chemotaxis equation, or the Keller-Siegel equations. So then in addition we have something for the signal. So the signal diffuses ds delta S. And then we have a kinetic term f of S and U, so that the signal is produced and also decays or can be consumed or something. So here often f of S and U is alpha U minus beta S. No, not beta. Let's say mu S. So just a linear production by U and linear decay with the rate mu. And so then this together here is called the Keller-Siegel equations. Or sometimes they are just called Keller-Siegel equations or chemotaxis equations. Now Patlak was actually the first to have these equations written down. And I don't know if anybody of you has read Patlak's paper. So have you read Patlak's paper? No. It is completely impossible to read. It is just so impossibly written, so convoluted, and the arguments go up and down and back and forth. I think it's from 57. It's very, very hard to read, but at some point the Keller-Siegel equations show up. So credit goes to Patlak as well. Actually, now I'm not sure about. I think it was 59 or something, and then Keller-Siegel was 1970. So to be on the safe side, we call it Patlak-Keller-Siegel model, and that's what it is. So I said this should be an application of what I did here with this measurement. So let's put this together. And let's just do it in 1D, because I'm lazy. So let's do 1D, then we have UT equals dUx minus beta UX. And I use the notation beta again. Beta is he times gradient of S. So maybe just write this down again. So beta is he times Sx. Okay. But this is not in this form, right? This is not in this form here. So let's bring it to this form so that we can really compare the terms. So then this is UT equals a d uxx minus beta xu minus beta ux. So now it's still not in the form, but almost. So we have one term too many, right? So we have ut is pt, cpx is this minus beta ux, dpxx is a duxx. But there's one term too many, which is this guy here. So let's set it to 0. So then in the middle. So if beta x is 0, then we can identify, so then beta is equal to c and d is equal to d, which was the expectation over time, and this was 1/2 the variance over time. So then, if we know beta x is 0, then we can make a direct relation to the measurements again. So we can inform the chemotaxis model from the population data which we might have available. So, yeah, to finish this off, I have a question to you. So I assume I can do this when beta is constant, basically, right? Beta x is 0 means beta is constant. But what if not? So what if not? So how is my question? So what if beta x is non-zero? So can I not relate any data to the model, or what would I have to do if beta is not a constant? So if you were my graduate student now for the next 10 minutes and I give you this question. So I said, I worked on this part here for like many years, and this is as far as I got, and now you should do it for beta x is non-zero. How do you relate it to the data? So this question is not easy. I do have an answer, but I would like that you think about this. So if you like, you can talk among yourselves. Just talk to your neighbor or something, or write something on a piece of paper and think about it.
So even if beta is not constant, what you get is beta and d.
Right, then I get this term here, right? This term here. I keep, yeah.
So the data you get is d.
What do you mean?
Your data.
From the data,
From the data you get the d and the speed.
D and C, I get these two, yeah.
So C corresponds to beta.
Right.
But can you see the C is a space function? Depends on space?
Yes, you're going in the right direction, yeah. So his idea was that C is spatially dependent. What does it mean for the random walks, which I studied? So coming back to your question, I don't know who asked it, one of you guys. Or did you ask it? Some of you asked me when Q is constant. Or Q is constant, you did this, right? Yeah. So that's it. If this beta depends on space, this means going back all the way, this Q depends on space. So at each location you have different probabilities of jumping left and right. So then you have Q of x, and you have to start all over again and do everything again, which is not very hard to do. But then you can resolve this problem. Yeah, okay. Very good. So then the answer is, so then Q is no longer constant. But depends on x. And this can all be done. Oh yeah, depends on S and Sx. That's really what it does. Then it really depends where the signal is and where you go up or down. Good, this is as far as I wanted to go today. So then I finish for now. We have a little break, and I see you tomorrow. Thank you.